Okay, so today we're diving into something pretty fundamental about how all life, even the simplest life forms, learn and adapt.
Yeah, really getting down to the basics.
Yeah. And the sources you shared lay out this fascinating theory.
Mm-hmm.
They call it a foundational theory for decentralized sensory learning.
Mm-hmm.
And it's a pretty wild ride, I got to say,
Yeah, definitely a thought-provoking one.
to put it mildly. So basically, the idea here
Yeah,
is that everything from like a bacterium sensing its environment
right.
to us, making decisions right now
Yeah.
is all rooted in this same basic principle
Exact.
of minimizing sensory input,
That's the cure idea.
which is
Yeah.
kind of a mind-blowing concept when you think about it.
Yeah, it's like a universal form of negative feedback that drives everything.
Yeah, it's like life is constantly trying to
Mm-hmm.
turn down the volume on all the signals it's receiving.
Yeah,
And by doing that, it figures out how to survive and thrive.
exactly.
So I can't we go any further. I think we got to clarify what we mean by sensory input here, right?
Absolutely.
Because we're not just talking about the five senses that we're familiar with.
No, not at all.
Like sight, sound, touch, taste, smell. It's much broader than that, right?
Much broader. So the researchers define a sensor in a really broad way. So
Okay.
it could be something internal, reflecting the cell's internal balance.
Right, like it's equilibrium.
Exactly, what we call homeostasis.
Mm-hmm.
But it could also be something external, like receptors on a cell's surface detecting changes in the environment.
So anything the cell can detect, basically.
Yeah, any input that can trigger a response.
Okay, so that's a pretty wide range of stuff.
Definitely covers a lot of ground.
Yeah. So the goal of this deep dive for our listeners is to really get a grasp on this whole evolutionary perspective.
Yeah, that's the key.
We want to explore this idea of sensory minimization
Mm-hmm.
and see how it could potentially explain all the amazing diversity of life on Earth.
And how it learns and adapts.
Right, and how it learns and adapts.
Yeah,
And we'll even connect this to how we understand our own brains,
absolutely.
and maybe even how we're developing artificial intelligence.
That's a fascinating connection.
Yeah, so it's a pretty ambitious goal.
It is, but I think it's a worthwhile one.
For sure. So the paper we're looking at,
Mm-hmm.
it draws insights from all across biology.
Yeah, it's really interdisciplinary.
From the very origins of life,
Right, the simplest organisms.
all the way up to the complexities of the nervous system.
The whole spectrum.
Yeah, so it's a pretty comprehensive view.
It offers a unified view of how learning might work at all levels.
Okay, so let's jump right into the heart of this theory.
All right.
The core idea, as we said, is that sensory signals,
Mm-hmm.
they're a problem.
Essentially, yes.
So
Yeah.
what's the thinking behind that?
So the basic idea is that every single cell, every single one,
Wow.
is constantly working to resolve imbalances
Okay,
by minimizing the signals it receives through its sensors.
okay.
And remember, these senses can be internal or external.
Right, right.
And the paper proposes that this ability to adapt and learn, this fundamental capacity, it's not something that emerged late in evolution
Okay,
with, complex organisms like us.
yeah.
It might have been present right from the very beginning.
From a get-go.
From the very first life
So
forms.
even a single-celled bacterium
Yeah.
has this inherent drive
Uh-huh.
to minimize signals.
That's the proposal, yes.
How does that even work in something so simple?
Well, think about the most basic needs of a cell.
Okay.
Things like getting energy, getting nutrients.
Uh-huh.
The paper argues that these basic needs,
Mm-hmm.
they drive internal sensors.
Okay.
Let me give you an example.
Yeah.
The sodium-potassium pump,
Okay.
which is crucial for maintaining a cell's internal environment.
Right, it's homeostasis.
Exactly.
So it's like the cell's housekeeper,
In a way, yeah.
keeping everything tidy and in
Mhm.
balance.
And that pump constantly needs energy to function.
Okay.
And that energy requirement can be seen as a primary problem the cell needs to solve.
So just existing, just maintaining itself
Right, just the basic upkeep of life.
is a problem that needs to be solved.
In a sense, yes.
Okay. So the cell is constantly dealing with these problems, and any disruption to this balance triggers these internal sensors
Okay, which then push the cell to take action
to fix it.
to restore that balance, yeah.
So even at this super basic level,
Yeah.
you see this drive to minimize a problematic signal.
Absolutely.
Okay, so I think our listeners would really benefit from a concrete example here.
Okay, sure.
And the paper talks about what happens
Mhm.
when a cell is in an environment that's lacking potassium ions.
Right, potassium is essential for many cellular processes. So imagine a cell that finds itself in a solution
Okay.
that doesn't have enough potassium.
Okay.
Now, the sodium-potassium pump,
Right, our little housekeeper.
exactly, it normally uses energy to pump sodium out
Mm-hmm.
and potassium in,
Okay.
but without enough potassium outside,
Mm.
the pump can't work as effectively.
Right, it's like trying to fill a leaky bucket
It can't keep up.
when there's not enough water in the, well,
Exactly.
so what happens then?
So there's always this slight leak of potassium out of the cell,
Okay.
which the pump normally corrects for.
Mm-hmm.
But now, without enough potassium outside,
Uh-huh.
the pump slows down,
Okay.
and the electrical charge across the cell membrane.
Right, that's called the membrane potential, right?
Exactly.
So that's like the cell's electrical
In
battery.
a sense, yeah.
That's what powers a lot of its functions.
Mhm.
So what happens to that battery when the pump slows down?
It starts to change. It becomes more positive inside.
Oh, interesting.
Yeah, this change in the electrical potential
Mm-hmm.
triggers these voltage-sensitive ion channels to
Like little gates in
exactly.
the cell membrane.
And this allows other ions, like calcium,
Mhm.
to rush into the cell.
So it's like a chain reaction.
Yeah, like a domino effect.
And this influx of calcium
Mhm.
sets off a whole cascade of internal reactions,
Absolutely.
activating all sorts of proteins and enzymes,
The cell is scrambling to respond.
trying to fix the problem.
Exactly.
So this cascade of reactions,
Yeah,
it's the cell's way of trying to fix that imbalance of ions.
absolutely. It's trying to restore equilibrium.
Okay, so the potassium deficiency,
Mhm.
that's the problem.
Right, the signal, the
The
problem.
signal, the problem.
Mhm.
And the cell's doing everything it can to fix it.
And this could even lead to changes in how it moves.
Oh, interesting.
Maybe directing it towards an area where you can get more potassium.
So even a single-celled organism
Yeah,
can exhibit behavior
in a way,
in
yeah.
response to these signals.
It's responding to its environment.
And the authors of the paper,
Mm-hmm.
they sum this up really nicely
Mm-hmm.
in their first conjecture,
Okay.
which is sensor signals integrated by a cell constitute problems which that cell needs to resolve.
The signal is the problem.
Right, it's not just information.
It's something that needs to be acted upon.
It's a call to action for the cell.
Absolutely.
Okay, so we've seen how the cell senses a problem
Mm-hmm.
and then tries to do something about it.
Right.
That leads us to the next question.
Okay.
How do cells actually respond?
Good question.
What are their tools?
Yeah, what's in their toolkit?
How do they link these problems to solutions?
So cells have a pretty diverse set of responses.
Okay.
They can adjust their internal metabolic processes.
Okay.
They can pump chemicals across their membranes.
Uh-huh.
They can even change their shape
Wow.
or move
Really?
using structures like cilia.
So they can physically rearrange themselves
Yeah, they're quite dynamic.
in response to these signals.
Exactly.
That's amazing.
And you can actually see some of this illustrated in Figure 1.
Oh yeah, figure 1 is really cool.
It shows this multifunctional cell
Mhm.
with all these different ways of sensing
Right, like light touch, chemicals,
all sorts of inputs.
and then all these ways of acting.
Like moving, secreting.
It's incredible how much a single cell can do.
Yeah, they're remarkably versatile.
So it's not just passively receiving information.
No, they're actively responding
It's interacting with its environment.
and adapting to it.
And adapting.
Yeah.
And these responses,
Mm-hmm.
they're not just fixed either.
No, they're not hardwired.
This can actually adapt them.
Based on their experience.
Oh, wow.
So changes in gene expression
Okay.
can lead to modifications in the cell's machinery.
So they can actually change their internal workings?
They can retool themselves.
Wow.
They can also regulate the number of sensors they have.
Oh, interesting.
So they can increase or decrease receptors on their surface.
So if they're encountering a certain signal a lot,
Mm,
they can become more or less sensitive to it.
exactly. They can tune their sensitivity.
That's pretty sophisticated for a single cell.
Yeah, it is.
It's like it's learning
in a rudimentary way, yeah.
from its experiences.
It's adapting its responses to be more effective.
So it's not conscious learning,
No, not in the way we think about it.
but it's still a form of learning.
It's adapting its behavior based on experience.
Right. And the paper captures this in 2 more conjectures.
Okay.
Conjecture 1.2,
Mm-hmm.
which says
Go ahead.
the cell couples problems, i.e. the sensor signals,
Right.
to responses which trigger some correction aiming to resolve those sensor signals.
So it's all about connecting problems to solutions.
Right. It's a feedback loop.
Absolutely.
And then conjecture 1.3.
yeah.
This one emphasizes the role of the cell membrane.
Okay.
It says, each membrane interface in a cell, such as a channel, cilia, or receptor,
Mm-hmm.
is evolved either as part of a sensor for some problems,
Right.
an actuator with which to resolve such problems, or both.
So the membrane is the interface
It's the boundary
between the cell and the world
and is actively involved.
in both detecting and solving the problems.
That's pretty remarkable.
It's the cell's front line.
So the cell's entire existence
Yeah.
is centered around this constant cycle of sensing and solving.
It's a fundamental principle of life at that level.
Okay, so we've talked a lot about single cells.
We have.
But obviously life is much more complex than that.
Oh, yeah.
We have multicellular organisms.
With trillions of cells.
With specialized cell types.
Working together.
So how does this theory scale up?
That's a great question.
What's the driving force behind all that complexity?
So the paper suggests that it's all about solving problems.
Okay.
Like getting food more efficiently. When you have different types of cells that specialize in different tasks, you can divide the labor.
Right, like an assembly line.
Exactly.
So you become much more effective.
As a whole organism.
But this specialization, it introduces a new challenge.
It does.
How do all these different cells communicate?
That's the key question.
How do they coordinate their actions?
Because now you have a situation where a sensory signal detected by one type of cell might need to be addressed by a completely different cell.
Right, locally to somewhere else in the body.
Exactly.
So you need a way to transmit those signals.
Yeah, those problem
from the right
signals
place.
to the cells that can actually do something about it.
And the paper shows this really well in figures 3A and B.
Right.
With these early forms of chemical signaling.
Yeah, the beginnings of intercellular communication.
It looks like these early multicellular organisms, they relied heavily on releasing chemicals.
Right, chemical messenger.
To talk to each other.
To coordinate their activities.
Which is fascinating because many of those chemical signals, like neuropeptides, are still used in our brains today.
As neurotransmitters.
It's like a continuation.
It's a remarkable continuity.
Of this same fundamental principle.
That we saw in single cells.
Where a sensory signal triggers this internal chemical problem signal.
And now, in multicellular organisms, these signals can be passed between cells.
Like a relay race.
Yeah, passing the problem
with the right team member.
To the cell that can solve it.
So it's like one cell senses a problem it can't handle, and it sends out this chemical SOS to another cell that can help.
They call in the specialists.
And the paper formalizes this with two more conjectures.
Right.
Conjecture 1.4 says, some cells can convert a sensed problem into a format detectable by another sensor.
So they're translating the problem.
Right, into a language other cells can understand.
Into a chemical language.
And then goes on to say, depending on the method, the converted problem signal can then be detected by the same cell.
Right.
Or by one or more remote cells.
So it can be a local or a long-distance call.
And then conjecture 1.5.
Yeah.
This one's really crucial. It says, a problem transmitted through a cell or between cells must not be discarded unless resolved.
So the problem persists.
It doesn't just disappear.
Until it's addressed.
The organism keeps working on it.
It's like a to-do list.
Until it's checked off.
And it can't just be ignored.
So that implies that the existence of multiple problems within an organism can lead to some pretty complex behaviors.
Absolutely.
Because trying to solve one problem might make another one worse.
Right. It's a balancing act.
Constant trade-off.
The organism is juggling priorities.
Okay, so this need for communication, especially rapid long-distance communication.
Yeah.
This seems to be where neurons come in.
Right, the specialized nerve cells.
How does the theory explain their emergence?
So the paper proposes that as multicellular organisms got bigger, relying solely on diffusion.
Right, that's where chemicals just spread out.
Yeah, it's slow
transmit signals.
It's not efficient over long distances.
So they needed something faster.
Exactly.
And that's where neurons evolved.
With their elongated structure.
Right, their long axons,
wires,
connecting distant parts of the body.
Enable a much faster signaling.
And we can actually see this in figure 3C.
Yeah, it shows how diffusion becomes limiting.
As distance
and neurons overcome that limitation.
So neurons are like a high-speed communication network.
For those problem signals.
So their primary role, initially at least, wasn't necessarily complex information processing.
Right, not the way we traditionally think about the brain.
It was more about relaying these basic problem signals.
Yeah, getting the message across quickly to the cells that can take action.
So it's still about problem solving.
At its core, yes.
Even at this level.
Even in the nervous system.
And synapses. Those junctions between neurons, they evolved as this really effective way to transmit these signals within neural networks.
To make those connections efficient and reliable.
And we see that in figure 3D.
Yes, showing the evolution of synapses.
Okay, so this leads to two really interesting conjectures about the brain.
Let's hear them.
Conjecture 1.6 says, foundational brain structures are not extracting information to evaluate and learn from, but communicating sensed problems efficiently in a common form from cell to cell, so that each detected problem is separated and resolved by a suitable actuation mechanism.
It's about distributing the work.
Dividing and conquering.
To solve the problems effectively.
And then conjecture 1.7.
Yeah.
Which follows from this. It says, states which arise in the brain are byproducts of the process described in conjecture 1.6.
So our thoughts, our feelings,
I was conscious of that.
They emerge from this underlying problem-solving activity.
That's a pretty radical idea.
It shifts our perspective on the brain.
It's not just a computer.
No, it's not just processing data.
It's a problem-solving network.
At its most fundamental level.
And that changes how we think about intelligence.
And consciousness.
Absolutely. So we've got this brain that's constantly receiving all this sensory information.
From all over the body.
From the outside world.
A constant stream of input.
So how does the organism decide what's important?
Right, that's the question.
What to prioritize?
How to allocate its resources.
And that brings us to the idea of sensory prioritization.
Absolutely. Not all signals are created equal.
Right. Some problems are more urgent than others.
Exactly.
So the paper gives some great examples.
Okay.
Like the buildup of carbon dioxide in your blood. That's a pretty immediate threat.
You need to breathe.
Whereas something like the subtle details in your peripheral vision.
Right.
That's not as critical.
Not for immediate survival.
So some inputs are more acute.
Yeah. They signal a more pressing problem.
They demand attention.
Like a loud alarm.
And it seems like there's a hierarchy of control involved here.
Okay.
Where more basic networks.
Like the spinal cord.
Right. The brainstem, they handle some of these less critical signals.
Automatically.
Without conscious involvement.
Keeping them at a baseline level.
Yeah. Maintaining the essentials.
And the paper points out that different sensors have different activation thresholds.
Right. Some need a stronger signal
to trigger a response.
To get their attention.
For example, the nerve endings that detect tissue damage.
Right. Beam receptors.
They only fire when the stimulus is strong enough.
To indicate a real threat.
So it takes a lot to set them off.
They're not easily triggered.
Which makes sense.
You don't want to be bothered by every little thing.
You need to focus on the real dangers.
The acute problems.
And you mentioned earlier how we might stop breathing briefly.
Right. When lifting something heavy or not notice a minor injury.
Yeah.
During intense exercise.
In the heat of the moment.
Those are great examples of how our sensory priorities can shift.
Based on what the body perceives as most important.
In that moment.
At that particular time.
So the context matters.
Absolutely.
And the authors sum this up in conjecture 1.8.
Okay.
Which is a bit of a mouthful.
Yeah.
But it basically says a problem signal in or between cells is comparable to any other problem signal.
Okay.
Regardless of origin, when in compatible chemical or electric form at the same location.
Right.
And the acuteness of such a problem is measurable in the magnitude of the signal.
So it's a strong signal.
It gets priority.
Whether it's internal or external.
Regardless of where it comes from.
Yeah.
And the paper gives all these everyday examples of this inaction.
Like what?
Like a blocked airway.
Right. That triggers an immediate response.
You can't breathe. It's a top priority. A newborn baby focused on hunger.
Gotta get fed.
Even just dryness in our eyes.
Yeah.
Triggering a blink.
To protect the eyes.
It's all about prioritizing.
Based on the acuteness of the signal.
The urgency of the problem.
Exactly.
And this extends to things like our sense of balance.
Right.
Kicking in when we start to fall.
To prevent injury.
How we adapt to muscle fatigue and pain. To avoid overexertion.
And even the possibility that certain nerve fibers in our ears have this high activation threshold to protect us from loud sounds.
So they only fire when the sound is really dangerously loud.
To prevent hearing damage.
So it's like a safety mechanism.
They're built into our biology.
It's fascinating how much of our behavior is driven by these underlying principles.
These basic drives.
To minimize these sensory problems.
That's the core idea.
So this whole concept of sensory minimization as problem solving, it reminds me a lot
of what?
Of the idea of feedback control.
Right.
That engineers use.
To regulate systems.
So how does this theory connect to that?
So the authors actually make an explicit connection to something called PID systems.
Proportional integral derivative.
Yeah, those are used in engineering to control all sorts of things.
Like thermostats.
Exactly.
And this principle of negative feedback control.
Which is illustrated in Figure 4.
Yeah, Figure 4 shows a nice diagram of that.
So think of it like your home thermostat.
Okay.
It senses the current temperature. That's the sensory input.
Okay.
And it compares that to your desired temperature setting.
Right. The set point.
Exactly.
The temperature you want.
And then it activates the heating or cooling system.
To minimize the difference.
To get the temperature back to where you want it.
So it's constantly adjusted.
To maintain that set point.
So in the context of this biological theory, every sensory signal has an ideal set point.
representing a state of minimal
Per optimal balance.
Yeah, like a perfect equilibrium.
And neural networks, they're primarily dealing with inputs that represent a deviation.
Right, an error.
From those ideal set points.
So they're constantly trying to correct those errors.
To get back to that optimal state.
That's the driving force.
And the authors even suggest that sensors we think of as purely informative.
Okay.
Like tactile sensors in our skin.
Right, that allow us to feel textures?
They might have this underlying connection to potential problems.
Like what?
Like the risk of cell damage from too much pressure.
So even feeling could be rooted in this drive to avoid harm?
That's really interesting.
It changes how we think about our senses.
It's not just about gathering information.
It's about protecting our data.
Maintaining our integrity.
At a fundamental level.
Okay, so how does all of this relate to existing theories of learning?
Yeah, good question.
Like Hebbian learning.
Right, the idea that cells that fire together wire together.
And spike timing-dependent plasticity.
Which refines those connections.
Based on the timing of neural activity.
Precisely.
So how does this sensory minimization framework fit in?
So the authors suggest that it can provide a new perspective on these well-established phenomena.
And neuroscience.
Yeah, like those you mentioned.
So it's not necessarily contradicting those theories.
Not necessarily.
It's offering a different lens.
To view them through.
A deeper level of explanation, perhaps.
And what about the concept of sensory prediction errors?
That's a hot topic in neuroscience these days.
Yeah, the idea that the brain is constantly making predictions about what it expects to sense.
And then comparing those predictions to reality.
And learning from the difference.
Right, the prediction error.
So how does that fit into this framework?
So the paper proposes that these prediction errors might naturally arise.
Okay.
From this sensory minimization principle.